Good afternoon. First of all, thank you for the opportunity. And just to introduce myself, my name is Thaise Gonçalves Araújo. I'm from Brazil, and I did my PhD in the Federal University of Uberlandia with Phage-display technology. And I worked at the NIH with this technology, improving antibodies against HIV. So today, I'm going to talk about peptide-displaying phage-display technology in breast cancer diagnosis, which was developed at the Federal University of Uberlandia at the Laboratory of Nanobiotechnology. This is one research line of our lab that use phage-display technology insert peptides, molecules, for clinical practice, both for diagnosis and for therapeutic purposes. In this afternoon, I'm going to talk a little bit about breast cancer, the phage-display technology, and explain how it works. The FABC4, this is an interesting recombinant antibody, this FAB, that we constructed, that I constructed with the co-authors during my PhD, and this recombinant antibody has interesting characteristics when we consider breast cancer. I'll talk about our objective, hypothesis, the methods, results, and the discussion and conclusion of our work. Just to talk a little bit breast cancer, and this is the second leading cause of death among women. And new cases will rise from 20 million in 2000 to 30 million in 2020. This is the second, this is the most common malignant tumor after non-melanoma. And epidemiological data from the National Institute of Cancer in Brazil shows that the mortality of this neoplasm is still alarming, even with the improvement of diagnosis methods. The estimated risk for this neoplasm in Brazil is about 57,112 new cases for 2013 and 2014. And for the Southwest, is about 71 new cases per 100 million women. The delay in diagnosis compromised the treatment of these patients. And most women, and a lot of women, still succumbs with a poor prognosis. The most important methods for diagnosis is the digital mammography that is done by women with 40 years old. So the search of the new biomarkers can open new possibilities for the treatment and for diagnosis of these neoplasms. And the discovery of new biomarkers and its molecular association with the occurrence and development may help in clinical practice and may help to understand the molecular, to understand the neoplastic phenotype. New biomarkers may help in diagnosis and prognosis, may help in therapy and response to therapy. There we usually say predictive biomarkers. We usually search for specific biomarkers, can help in classification. In breast tumor, we have the molecular classification, and our FABC4 open new possibilities for this molecular classification and the early detection of malignant phenotypes. So the search of these new biomarkers has recently relied on phage-display technology. This technology is successful in select peptides or proteins against a specific target from a vast variance of peptides. So the phage library, we work with a library, and the phage library display different peptides. It is exposed to a target, and phages with appropriate specificity are captured. Non-binding phages are washed off, and bounded phages are eluted. We have different ways to do this elution. Usually use acid solution or molecules that compete for this target. So the eluted phages infect host bacteria and are therapy amplified. After 2 to 5 rounds of selection, we have monoclonal peptides that can be analyzed individually. So considering tumor and consider breast cancer and breast tumor, this is a useful technology because breast cancer is a multifactorial, heterogeneous, and multifocal disease. And this kind of technology may help us to select and to consider this molecular diversity of breast cancer. Using this kind of technology, we constructed the FABC4. The FABC4 is a recombinant antibody and that could differentiate tissues from breast cancer tissues, from benign breast disease with no malignant transformation or no malignant phenotype, and normal tissues. So it was able to differentiate these 3 groups. And moreover, it was also specific to breast cancer because FABC4 did not recognize prostate cancer, pancreas, this is stomach, and lymphoma. So it can be used for diagnosis when we consider biopsies, and it's specific to breast cancer. The most interesting result that we have for this FABC4 is that it could differentiate patients with different outcomes when we consider triple negative breast cancer patients. This is an extremely heterogeneous group of patients. That is, there is no kind of treatment, adjuvant treatment. So we can't treat this patient with hormones or with antibodies. It's only chemotherapy and radiotherapy. So it's an extremely heterogeneous group of patients. Our FABC4 could differentiate this kind of patients with different outcomes. So when the patient has the antigens that is recognized by FABC4, they present higher disease-free survival and overall survival. When you consider disease-free survival is about 74 months when positive, and only 13 months when negative. And for overall survival, it was about 84 months when positive for FABC4, and only 21 months when negative for FABC4. However, its target, its antigen, it was not yet characterized. Based on that, we aimed with this study to identify a new marker for breast cancer diagnosis which recognize FABC4. So we are trying to identify the antigen that is recognized by FABC4. Our hypothesis is that mimotopes peptides specific for FABC4 are able to discriminate breast cancer from benign and normal sera. This is a diagnosis objective, but we also have perspectives for therapeutic purposes. Based on that, what we did, we subcloned FABC4 in non-suppressor strain of E. coli, and we induced FABC4 by IPTG for HPLC purification. Purified FABC4 was used in a biopanning assay. This is the selection procedure in Phage-display. So purified FABC4 was used as target for phage particles. So a library, PHD12 library phage, was exposed to FABC4. To isolate phages specific to FABC4, we performed a subtractive selection with an irrelevant FAB. Phages that recognize this irrelevant FAB were not considered, so we used just the supernatant with non-adhered phages, which was submitted to a positive selection against FABC4. The elution was performed in 2 steps. First, we used proteins from benign patients, proteins from benign tissues, which was discarded. And the second step with proteins from breast cancer tissues, which was amplified, which was used to infect host bacteria and was amplified. After 3 rounds of selection, we analyzed individually all 96 clones by different assays, and we performed the other procedure to validate these phages. One of this validation was done by ELISA, and we separate our patients in 3 groups, to 3 different groups. The first one, the breast cancer groups, that included in situ carcinoma, invasive ductal carcinoma. The median age was 46.2 years old. The majority of our patients was T2 and D2. This is histopathological data. We also analyzed their hormonal status and for HER2, if they have or not amplification for HER2. For benign breast disease, we analyzed and we grouped the patients with fibroadenoma, papilloma, cyst, ductal hyperplasia, and the median age was 46.8 years old. And finally, the normal group was patients that were submitted only through mammoplasty with no alteration in the breast. The first question that we have to answer when we are working with phage technology, if our biopanning or selection process was success. So after the 3rd round of selection, we picked up 96 clones, that is clones. And our biopanning process was successful because we could select 6 clones that recognize FABC4 that presented higher absorbance when we consider FABC4 comparing to the irrelevant FAB. So we have, in the end of the process, by Phage-ELISA screening, 6 promised clones. These 6 promised clones were tested against pooled IgG, this part of the graph. So when we analyze these 6 promised clones, only 4, A5, A7, C4, and D6, were able to discriminate breast cancer and benign disease. The other A7 B12 only discriminate benign disease and normal, and D12 did not differentiate our groups. So after that, we analyzed these 4 clones individually when we consider the sera. We collected 50 patients, sera from 50 patients, and we analyzed this sera against these 4 promised clones. And they, again, differentiate breast cancer from benign disease and control, and the normal sera. After that, we construct ROC curve for analyze the diagnosis, capability of these clones. And the area under the curve was about 0.7. The sensibility was about 50%, and specificity was about 85%. And finally, we synthesized chemically these 4 clones, but only 2 peptides were able to be recognized by FABC4. Among these two, only these 6, the peptide, the chemical peptide, these 6 were able to discriminate our 3 groups. So we are working now on with these 6, studying it could discriminate the 3 groups, and we analyze the 6 as a therapeutic molecule. So, triple-negative breast cancer represents a subset of aggressive tumor, account for 15 to 20% of newly diagnosed breast cancer cases, and this group is recognized by FABC4. Comprehension of its heterogeneity open new possibilities of treatment, and our results leads to a conceptual shift in the diagnostic and prognostic of breast cancer. Human disease is often caused and associated with alteration in protein expression, and this alteration can be characterized by phage display. The identification of new marker or putative signaling pathways may allow the design of new diagnostic and therapeutic strategies. And in summary, our D6 phage open new insights for clinical practice, and it could be applied in clinical trials as a complementary method to mammography. Phage-ELISA is a reproducible, simple, fast, and inexpensive method. And phage display is a successful technology considering breast cancer. Finally, I would like to thank Federal University of Uberlandia, the co-authors of this work, the Professor Luiz Ricardo Goulart, Yara Cristina de Paiva Maia, Vanessa da Silva Ribeiro, Emília Rezende Vaz e Galber, the University Hospital with Donizete, Luanda e Tânia, Barretos Cancer Hospital with Carlos Eduardo Paiva, and AC Camargo Cancer Center with Fernando Soares and Rafael Malagoli. Thank you.
Very nice talk. Enjoyed it very much. Where exactly is your protein located? The protein that's...
Which protein you mean? FABC4?
That you found in your as your new marker.
Oh, the peptide that I selected against FABC4. Well, we sequenced the DNA, and we are analyzing the peptides. So we have 5 different sequences. And we analyze this. In a first assay to discover and to characterize the antigen that is recognized by FABC4, we performed mass spectrometry, and we identified cytokeratin 10. So cytokeratin 10 is in the cytoplasm. So, but we know that it's not only one epitope. We think that it's a kind of interaction of different proteins that is recognized by FABC4. That's why we performed the phage display. If we're able to capture this epitope with different proteins. But in a prior experiment, we identified cytokeratin 10 that is in cytoplasm. And FABC4 is able to go through the membrane and identify something in the cytoplasm.
So, but you don't know yet whether this is the only sequence. I mean, you have...
No, we didn't know yet. It's really difficult, and it's a promise marker when we consider FABC4 because of the triple-negative breast cancer group. We don't have anything to treat this group, only chemotherapy and radiotherapy. So it's really, really important to characterize it.
Right, exactly. Okay, thank you.
You're welcome.
Any more questions? Could I ask one, perhaps? When it comes to cancer, you are looking at the primary tumor. So there's a twofold question here. The first question is, does it also work against the secondaries? And the second is, can you therefore also use circulating tumor cells as an opportunity to identify relapse, for example, after primary treatment?
You mean the second primary tumor, or you mean metastasis?
Metastasis.
We didn't analyze lymph nodes, and we didn't analyze. We just know that our FABC4 is able to discriminate different outcomes when you say disease-free survival or overall survival. But we did not analyze lymph nodes. We don't know if there is an alteration that is recognized by FABC4 in the lymph nodes. We don't know that.
Thank you. Okay, we have to move on. So,
okay.
Thank you very much.
Thank you.